Hi, my name is Chaitanya. I'm a graduate student in Mona Singh's lab at Princeton University, and I'm going to talk to you today about methods we're developing for identifying evolutionary origins of repeat domains. As many of you probably know, domains are structural and functional subunits of proteins, and almost all proteins contain at least one domain. What's interesting for us is that about 20% of all human proteins contain consecutive repeats of the same domain. So on the right, I have a depiction of a protein as a string, and domains are represented as shapes on that string. The consecutive repeats of the orange boxes at the end represent protein domain repeats. So these repeats typically work together to achieve function, and changes in domain repertoire through domain duplications and losses are a primary driver of protein evolution. Domain repeats occur in thousands of proteins across many subfamilies. And here's a look at several common repeat domains. They're involved in a lot of functions, including protein-protein interactions, DNA binding, signaling pathways, and many others. Let's take a look at a specific example, the cis2 his2 zinc finger family of domains. Zinc fingers are DNA binding domains. And what's interesting about this family is that the domains work together in triplets to specify a DNA binding site. So it'd be interesting to know whether evolution follows function in this case. So do these domains also duplicate in groups of 3? So it turns out that they do, at least in some cases. So here we have a zinc finger protein with 15 repeat domains. I plotted a pairwise similarity map of these domains, and we see that domains 1 through 3 are most similar to domains 4, 5, and 6, respectively. This is representative of a tandem duplication event in which multiple domains duplicate simultaneously rather than as individual duplications. I'd like to mention here that although we can sometimes identify tandem duplications in sequence like this, it's not always as clear, and we can't identify when in the evolutionary history of the protein these duplications occurred. So these are the types of questions that we like to answer with our framework. For the purposes of this talk, a tandem duplication is a simultaneous duplication of 2 or more consecutive domains. We assume that the duplicate copy is placed immediately after the original. So on the left, we have a protein which had 3 domains initially, in which the first 2 domains undergo a tandem duplication event. The 2 duplicates are inserted immediately after the original. The scenario on the right is not allowed in our framework because the duplicates are placed further downstream on the protein. In the rest of this talk, I'll first give an overview of the domain evolution process and previous approaches used to find tandem duplications. I'll then introduce our tandem duplication loss framework and then present our solution to the problem. Finally, I'll show some results on both synthetic and real data sets. So here is a representation of what domain evolution looks like in the context of gene evolution. So we have a simple representation of a gene phylogeny composed of 3 existing genes, 1, 2, and 3, and ancestral genes 4 and 5. Suppose that the ancestral gene contained 1 instance of a protein domain, shown as the black dot at the top. Any evolutionary event that occurs at the gene level also occurs at the domain level. So when you see a bifurcation in the gene tree, you see a corresponding co-duplication event in the domain tree. Domains can also duplicate within a gene. So the green dot at internal node domain node B indicates that that domain has been duplicated. And after that event, gene sequence 4 contains 2 copies of the domain, and that's represented by nodes D and E. Both of these domains are carried down to each of the gene's children. Along the way, we might lose a domain in the sequence, which I'm indicating with that blue X. And after the sequence of events, gene 1 is left with 1 domain, F, while gene 2 contains 2 domains, G and H. Finally, we can have a cascade of duplications, such as on the right. I've circled 2 domains here that might have been involved in a tandem duplication event. It is important to note here that we don't know from the tree alone whether this was actually a tandem duplication event or if it was 2 single duplication events. The typical process through which domain-level evolutionary events are inferred is called reconciliation. So in reconciliation, the input is a gene tree and a domain tree, and topological inconsistencies are reconciled between these two trees to find an event history and a mapping of internal domain nodes to gene nodes. Reconciliation captures domain-level events through a pre-specified evolutionary model. So that model typically includes events such as duplications and losses. And sometimes they include other events like transfers, merges, and splits. Most methods use a parsimony model, assigning cost to each evolutionary event and trying to minimize the cost of an assignment subject to biological constraints from the gene tree. In most cases, events are assigned to individual domains in the tree, which means that they can't capture tandem duplication events because those affect multiple domains at once. So to handle tandem duplications, several approaches have been proposed. And I'll call these models segmental duplication approaches. So the idea here is to map domain nodes to gene nodes, but then instead of having individual duplications, you group multiple domains into a single duplication event. Each duplication set is then treated as a single event. And the goal becomes to minimize the number of duplication sets in a reconciliation. So the issue with this approach is that it can't distinguish between a single tandem duplication event or multiple single duplication events. And I'll show an example of this issue on the next slide. So here we have a single protein with 3 domains, A, B, and C initially. And on the left and right, we have 2 different evolutionary scenarios for this protein. So on the left, we have a single duplication event in which all 3 domains are duplicated at once. The result is shown below that with 6 domains in the final protein. The domain tree that you build from these 6 domains is shown at the bottom left. On the right, we have the same starting protein, but instead of a single duplication of 3 domains, we have 3 individual duplications of size 1. The resulting protein still has 2 of each type of domain, and therefore the resulting domain tree is going to be indistinguishable from the previous case. Segmental duplication frameworks can't distinguish between these 2 cases because the domain tree topologies are identical. So what does change in these 2 scenarios is the final position of the domains on the protein. So we can augment the domain trees with these positions, which I've shown in red, to distinguish between the 2 cases. So on the left, you can see that the positions on the leaf pairs are interleaved. So here, this leaf pair has a 1 and a 4, and that's interleaved with the 2 and 5 in the next leaf pair, which is interleaved with the 3 and 6 in the final leaf pair. On the other hand, on the right, you can see that you don't have that interleaving pattern. So you can use these positions to distinguish between the 2 evolutionary scenarios. So here's our tandem duplication loss reconciliation framework. Our goal is to obtain an optimal reconciliation between gene and domain tree with a full history of tandem duplications and individual losses. Our inputs include the usual inputs, a gene tree and a domain tree, but we also include a pairwise eligibility matrix. So this matrix uses the position information I described previously to determine whether any pair of domains could have been involved in a tandem duplication event. Our output is going to be a full mapping of domains to genes, as well as an event assignment for all domains. We use a parsimony approach, so we also output a reconciliation cost for our reconciliations. So we have 5 main biological constraints. The first is that domains must evolve in the same direction as genes. So we don't allow for transfers in our model, so domains can't hop from one gene to another, as in that second picture. Our second constraint is that children of co-duplication nodes must be in separate lineages. So I'll note that the mapping in the example marked as incorrect is actually possible, but a domain would have to be assigned a duplication event there rather than a co-duplication event. The next three constraints have to do with duplication sets. So constraint 3 says that no domain in a group can be an ancestor of another domain in that same group. The fourth constraint says that all domains in a duplication set must be mapped to the same gene. And finally, the fifth constraint says that two duplication groups can't have conflicting ancestry relationships. So the idea here is that all domains in a group are inferred to have existed at the same time. So if you have groups that cross, as in the incorrect example on the right, then you have that conflicts with the ancestry relationships given by the tree itself. So I mentioned that we use a pairwise eligibility matrix to determine whether two nodes could be part of the same tandem duplication set or not. We say that two nodes in the domain tree are pairwise eligible if the positions of their children are interleaved. For leaf domains, this makes sense because we have position information from existing sequences. But for internal nodes, this idea becomes a little more complicated. We don't know what domains existed at the same time for internal nodes, and we don't even know whether they existed in the same gene or not. So to infer position-like information for internal nodes, we take the simple approach. And here we're going to take the minimum position value of a leaf in that node subtree and use that for pairwise eligibility computation. So we have two solutions to our reconciliation problem, an exact ILP and a faster heuristic. Our ILP solution takes gene and domain trees, as well as a pairwise eligibility matrix for input. Our output is an optimal mapping and a reconciliation cost, as well as all tandem duplication sets and losses that occur. So this ILP solution is exact and it works well, but unfortunately requires a large number of constraints and therefore doesn't scale well to larger families. To solve this issue, we also have a heuristic solution. The idea here is that domain duplications can only occur within a gene. So if we knew the optimal mapping of domains to genes ahead of time, we could break the tandem duplication identification problem into smaller subproblems. Rather than looking at the entire domain tree at once, we only need to look at domains that map to a particular gene. So that's basically what we do. We first get an imperfect mapping of domains to genes. And once we have this mapping, we take portions of the domain tree mapped to each gene separately. And we use a modified version of our ILP to identify tandem duplications within each of these genes. After this, we greedily remap domains to alternate genes. When doing so, improves our reconciliation. So here's a closer look at how these three steps work. So for our imperfect mapping, we use the simplest possible mapping that satisfies problem constraints. And that's going to be the lowest common ancestor mapping. So we know which leaf domains map to which leaf genes because we have those existing sequences. But for any internal domain node, we're going to map it to the gene that is the lowest common ancestor of the genes that its children are mapped to. So for example, if you look at domains 1 and 2, they're mapped to genes A and C respectively. So the ancestor of 1 and 2 has to be mapped to the root of the gene tree. Similarly, the ancestor of domains 3 and 4 maps to the lowest common ancestor of genes A and B. And we continue this process until all domains are mapped. So this mapping ignores tandem duplications entirely. But as we'll see later, it actually, our heuristic performs well despite this. So once we have our lowest common ancestor mapping, we need to find tandem duplication sets within each gene. So here we're going to use a simplified version of our ILP to get the lowest cost assignment of tandem duplications. So our simple ILP does not consider mapping constraints, and it only needs to look at a subset of domains to find these tandem duplications, so it's much faster than the full problem. So we start at the leaf genes, running our ILP at each of the leaves, and then work our way up the tree, looking at one gene at a time. And at the end, we have a full tandem duplication history for all domains and all genes in the tree. The last step of the heuristic is the remapping step. And here we use a greedy approach where we change the mapping of a domain when doing so reduces our parsimony cost. In order for that to happen, the number of duplications has to go down as a result of the remapping. So in this case, you can see that before remapping on the left, we had a domain here mapped to this gene, the bottom left gene. And by remapping it to the root gene, we can actually, in some cases, depending on the positions of these leaf domains, you can create a single tandem duplication event up here rather than having individual events, duplication events, as we had before. So we make two assumptions in our model. The first is that duplicated units are placed immediately after the original. And the second is that we assume that domain shuffling does not occur. So that means if you have a domain that existed before another domain in a sequence, those two positions will never swap later on. So under those assumptions, and assuming that no losses follow a tandem duplication, we can show that if a tandem duplication occurred to certain domains in a protein, then the internal nodes in the domain tree corresponding to those domains will be pairwise eligible. And that means that we can identify that tandem duplication. So although we do have this loss assumption, we find in practice that most losses do not hurt our ability to detect tandem duplications, as we'll see later. It's also important to note that the reverse is not necessarily true. So if you have two domains that are pairwise eligible, it doesn't mean that they were involved in a tandem duplication event. So you can think of our model as restricting false positive tandem duplication sets. So lastly, I'll talk a bit about our testing framework. Testing was done mainly in simulation, and we built a simulation setup in which we generate both gene and domain phylogenies. The gene trees are initialized as random topologies with a fixed number of leaves, and here we usually settled on 8 leaves. Branch lengths are assigned to be roughly even between edges on a root to leaf path, with small variance added in. We generate domain trees within this gene tree, starting with a single domain at the top and in the root of the gene tree. Co-duplication events occur in line with the gene tree, and between these events, we have duplications and losses that occur according to a birth-death process. The number of events on a root to leaf path is tuned using an event distance parameter. And here, lower event distances correspond to a larger number of events, whereas higher event distances correspond to a lower number of events. So we tested the accuracy of 3 different methods on identifying tandem duplications in our simulations. So the ILP and our heuristic, represented by green and orange bars, are the two methods that, the two solutions that we have to our tandem duplication reconciliation problem. The blue bar is for a method called multirec, which solves a segmental duplication problem. And here again, it's trying to find groups of duplication nodes, but without using the position information that we use. So as you can see, at lower event distances, our methods have slightly lower recall than the segmental duplication approach. But on the other hand, we have much higher precision at those lower event distances. And that indicates that we're able to correctly distinguish between tandem duplication events and repeated single duplication events. The other thing to note here is that our heuristic performs nearly indistinguishably from our ILP, which is a good sign. So here I'm showing the F1 score, which is a way to measure the trade-off between precision and recall. And as you might expect, based on the previous 2 charts, we can see that the F1 scores of our 2 methods are significantly higher than multirec at low event distances. So here I'm showing the runtime savings we get from using our heuristic. And we're comparing our heuristic again, both against running the full ILP and against running multirec, the segmental duplication approach. So the full ILP scales poorly at smaller event distances, and we were not actually able to complete within, I think, 24 hours at the lowest event distance. However, our heuristic takes about 4 seconds per problem on average, which is perfectly feasible. And we also show significant improvement in accuracy over Multrek while taking only a minor hit in runtime. I think it's also worth mentioning that if you have large gene trees, which can be common, our heuristic scales linearly with the size of the gene tree, which is another nice property to have. So for a real world example, we tested we examined 18 filament A proteins across 18 species that were previously studied. And based on heat maps of domain similarity sequence, that study concluded that there were lineage specific tandem duplication patterns in 2 of the species here, Trichoplex otorens and Hirudo medicinalis. So here I'm showing a species tree of the 18 species that were involved. And because these genes were part of a 1-to-1 orthogroup, we used the species tree as our gene tree. And the domain tree was built by extracting domains from all of these 18 sequences and comprised a total of 435 domains. And we built this tree using RAXML. So we then identified tandem duplications across the domain tree using our heuristic. So shown here are lineage-specific domain expansions for the 2 species of interest, Hirudo medicinalis and Trichoplex otorens. So the gray boxes indicate tandem duplication events, while all other nodes in each of these trees represent single duplication events. So on the left, you see a series of repeated single duplications, while on the right, we infer a few large tandem duplications. So our methods here are able to distinguish between these 2, while previous reconciliation methods cannot. So there are several avenues for further exploration. I think it'd be useful to incorporate multi-domain losses into our model, and other events, such as transfers, merges, splits, or domain shuffling, could be useful as well, depending on the domain or domain family of interest. And we'd also like to conduct large-scale surveys of orthogroups to determine how common these tandem duplication events are among various domain families, starting with the Zingfinger family. And so with that, I'd like to thank Mona for all our very helpful feedback and the rest of the Sing Lab for their insights. And I'm happy to take any questions you might have.